My name is Damon Ty. I work for Bio-Rad Laboratories as a curriculum and training specialist. And today's workshop, we're going to look at a lab for doing photosynthesis and cellular respiration, basically within the same lab system, which is kind of nice because it gets rid of some of the misconceptions that students walk away from a lot of times, which is that animals are the only ones to do cellular respiration. We constantly see this on like AP biology exams and things like that. So it's half the reason that we built this specific lab activity. I'm gonna go ahead and play a video for you. We're gonna have the chat going in the side. And so I'm gonna be on that. And then also George, who's one of our R&D folks at Bio-Rad, will also be in the chat. And so we can handle any questions that you've got in there. The video plays for about 30 minutes. And so we've got at least a solid 10 minutes at the end to do kind of more live kind of questions and stuff like that. But like I said, drop those questions as they come up in the chat and we'll try to deal with them there. All right, and with that, let me go ahead and do my screen share and I'll bring up this video NSTA had us pre-record. Second, what's going on here?
And welcome to our presentation.
Actually, give me one more second. Zoom doesn't seem to be finding that screen. I guess you guys will just get to see my full screen then. Let's do that.
Investigate photosynthesis and cellular respiration with algae beans. My name is Lisa, and I am a curriculum and training specialist for the Explorer program at Bio-Rad Laboratories. For more information about this lab, or for any questions about the Bio-Rad Explorer program, please email us at explorer@bio-rad.com and check out the links in the description. Feel free to also check out our Photosynthesis and Cellular Respiration YouTube playlist, where you can access 4 of our instructional videos that are designed to walk you, the teacher, through the prep stage and your students through the kids' core lab, including explanations of foundational content like the endosymbiotic theory and review of chemical equations that represent these processes to maximize for the kids' educational benefit. In this presentation, we will take a closer look at the kids' contents and model system, the kids' specific connections to select learning objectives and scientific practices within the College Board's AP Biology framework, instructor lab, the student core lab, inquiry-based extensions of that core lab, and information on how to purchase this kit. The Bio-Rad Explorer program offers a suite of hands-on experiences designed to promote critical thinking, develop reasoning skills, and to drive inquiry learning in the AP Biology classroom. As a part of this kit family, the Photosynthesis and Cellular Respiration Kit extends these scientific practices to your students while supporting their enduring understanding of cellular energetics concepts. The biological models utilized in this kit are live, encapsulated, single-celled algae, specifically Synodesmus obliquus. These self-sustaining algal bead units offer great value to both instructional and learning activities. This means that these beads, after given sufficient rest to downregulate photosynthesis, can be used from one class to the next. Algae beads are easy to maintain and are common in the prep. These beads can be stored at room temperature in a scientific container solution or in the fridge for longer periods of time between classes. Algae beads are easy to dispose. You simply need to treat them with some Leigh solution before disposing of the complete. Think about it. have taught your students about photosynthesis and cellular respiration. How many of them tend to remember that plants do both photosynthesis and cellular respiration? How many of them seem to understand the connection between these two cellular processes? Or do they simply see these events as signs and different mechanisms? One of the great aspects of photosynthesis and cellular which focuses on cell energetics, and then idea number 4, which focuses on system interactions. Now, the one at the core of that, the cycle to maintain balance within these systems, thus providing engaging learning support for multiple learning objectives and scientific practices within the biology curriculum. Two key benefits of this kit for instructors is its ease of prep and temperament qualities. This part of the experience can be simplified and described in a few easy steps. 1. Preparing your 1x out of an exam indicator solution. You can do this by simply writing the spell from an exam indicator solution provided in the kit and allowing it to incorporate to the exam's natural notes or measurement. 2. Wash and acclimate your large beans. 24 to 48 hours prior to cleaning the quarterback. Wash your hands and activate them and put 1x indicator solution on the back steps. A 16-hour position for the line to make the centimeters of the body inside the 3. Preps and workstations.
Hey everybody, so we're having audio issues. Can somebody confirm how well you can hear or not? Because I'm not hearing the audio very well at all. So if you can just put in the chat or unmute yourself, I'd appreciate that. What's the audio like for you guys? That's in and out. So we can just do a little more of a round table, talk about algae beads, give you some resources there. Does that work for you guys? Perfect. So Damon, if you're okay with that, we can just talk a little about what algae beads are and how they're, really helpful for conveying multiple aspects of respiration and how, they really, what's really wonderful about these algae beads is that it really encapsulates both sides of, photosynthesis, where you have respiration and you have the ability to, show these through pH changes, this wonderful indicator. And Damon actually has that wonderful picture right behind him of these algae beads. It's good stuff. Damon, do you think you can find another slide deck?
I'm trying to find one right now. The only problem is I'm having some other kind of computer problems. So give me one more second here.
No worries. You guys can feel free to unmute yourself and ask a question, or you can put it in the chat. What are you hoping to get from this session?
Actually, here we go. Okay, so I've got a slide deck that I typically use to teach us more of an ecological kind of context. So I'm going to take you guys through that since I'm not sure what's going on with the audio with the video we have. So like I said earlier, half the reason we built this kit was because we were finding that students were walking out of, especially we're looking at AP biology test scores, because that should give you a good pulse on kind of like what's going on and supposedly your best and brightest students coming out of biology in high school, and also should give you a rough estimate of what's going on at probably the starting levels of college. When we were looking at those test scores, we were finding that about 50% of the students walk out of AP biology thinking plants don't do cellular respiration. And so for us, we were like, how's that possible? Because you definitely get that in the theory portion of the course, But then if you look at the labs, a lot of people have kind of inherited or are running basically two separate labs to then examine photosynthesis and to examine cellular respiration. And so we thought, well, maybe that's the problem right there. Maybe it's the fact that we've got kind of two things that they're physically doing, and that's what they end up remembering. So how can we go about finding a way to get these concepts both in the same model organism? And so we spent quite a few years kind of figuring out how to do that. And what we came up with was basically taking a freshwater algae, Senodesmus obliquus, and encapsulating it into a biopolymer that made it easy to move basically a discrete number of algal cells around. And the other benefit to that was if you wanted to get quantitative, since the algal cells are now encapsulated in this biopolymer, you could also say drop them into something and you won't have all those cells in there. So you can actually use spectrophotometry to then look at it. So the other part of this system that this hinges upon is a really tightly built, basically kind of pH carbon dioxide indicator. And so with this, what's going to take place is you can drop these beads more or less in the pH indicator. And as the algal cells are doing different functions, they'll slightly shift the pH, and the students will be able to read that based upon a simple color change. So if you drop the cells in the pH indicator and you put them in the dark, those photosynthetic cells now are just doing cellular respiration. And so they're dumping CO2 out into the solution, and that solution will slowly kind of shift down in the scale and eventually get you to kind of a really bright yellow color. However, though, if you take those same cells, the same algae beads, and place them in the light, they're still doing cellular respiration right in the background. You got to do that in order to live, but they're also doing photosynthesis at this really high rate, which means they're actually gobbling up a lot of that carbon that got dumped out into solution. And they're also dumping O2 out into the solution. So you'll slowly get basically a pH shift upwards, and you'll get to this kind of purplish color. So actually in the beads behind me, whoops, backwards in here, you can kind of see that. So those are ones that have just come out of the dark and are being put in the light. And you can see kind of that purple aura coming from around them. And so that's basically them gobbling up that carbon, dumping the O2, and doing a slight pH change right around those beads. So the beauty about this system is it can take place relatively fast. So this can shift from, if you start like around 7.9, 8.1, this will shift all the way down to 6.9 in about 15 minutes and up to about 9.1, actually closer to like 10 minutes, depending upon your light source. And so the beauty is that it's a really fast system also to use in the classroom. And then being a former high school teacher, what I really like about it is I don't really like destroy anything in this. So I can really kind of like reuse these stations multiple times. And then Bio-Rad with this kit, they give you enough. Bio-Rad designs most of their kits to try to give you enough for 8 lab stations, usually with the idea of like 3 to 4 students around there. But they usually give you a lot extra. And so with this, you've got enough beads easily to make 8 lab stations. I can usually make 12 lab stations out of a kit. And each lab station is basically a cuvette that's going to go in the light, a cuvette that's going to go in the dark with 10 beads in it each, and then a milliliter of that pH indicator over the top. They give you 500 milliliters that pH indicator. And so that's, I mean, way more than you need for just one section. So you can usually just buy one of these kits and then cycle it through multiple sections, which is really nice. However, though, Bio-Rad a lot of times pitches these out there without having much context for why students might be interested in the technique. So I actually find that this, now that this lab is out there, and I feel that it makes photosynthesis and cellular respiration easy, it gives me more time to think about why do I want students to really understand photosynthesis and cellular respiration, especially at a rate level. And that's one of the beauties of this lab is you can really get nice tight rate studies with how quickly is it shifting towards the different ends of the color spectrum. So a lot of times I'll start off with an ecological context. It's best if you can find, of course, a local version of this, but I'll give you the one that we usually give at national shows just because everyone's very aware of this problem going on in the Gulf. And so I'll usually show students this photograph and be like, hey, first, what do you see? What's going on there? And kids will be like, a bunch of dead fish. And you're like, okay, good, all right. What's that area look like? And they'll make some guesses like, oh, it looks like a lake, looks like this, that. And then we'll ask, well, what might be causing like a big fish die-off like this? So kids will put out all kinds of crazy things. Usually, the first one that comes out is they'll just say pollutants. And you're like, okay, let's dive a little deeper. What do you mean by pollutants? And somebody might say, oh, I think they're they're putting out, soap in the water. Okay, yeah, that might be a possibility. Oh, they're putting out, runoff from a cattle farm. Okay, well, why is that? Why might that be a problem? And try and kind of dig out some of those threads and get them by hypotheses about why those fish might be there. And then what I'll usually do is add another layer of information to that and be like, okay, so actually what we're looking at is back here at the backside of Atchafalaya Bay. And so if you're not really good with your geography, we back out one more time and be like, okay, so we're actually at the bottom of Louisiana. Right over there is where the Mississippi River is kind of dumping out. And what do you see in the water there? We see a lot of basically sediment. And then, depending on how your screen is, you can actually see that there's also this kind of orange glow going out into the Gulf after that sediment kind of brown layer is stopped. And you can ask them, well, what's that from? If one of those kids gave you one of those threads of a pollutant being, run off from agriculture, you can kind of dig down a little bit deeper here and be like, okay, so that bloom we might be seeing, that might be from some of those agricultural outputs, right? So those are there to help plants grow, or from cattle feces. So you have all these nutrients flowing into the Gulf there, and we have this big kind of green color going on from that. What do you think that is? Usually you can get one kid to come up with the fact that it's some sort of photosynthetic organism like an alga. And then you can go, well, but why is that a problem? Why might that be connected to our fish over there? Because photosynthetic organisms, things eat those, right? Primary producers, isn't that a good thing? And usually it is, but there's also other effects because you have all those primary producers and they're doing primary production during the day. As they die though, they're falling down to the bottom, creating food for methanogen producers. But then also at night, all of those algal cells are switching over from producing oxygen to gobbling up oxygen. And so you can get these hypoxic zones, these oxygen, kind of dead zones that you see in the Gulf of Mexico. And there's a whole bunch of examples of this all across the United States. This one has, of course, the best resources, case studies, and things like that you can find all over the place. But I'd really encourage you to try and find one locally to use as a context for why we should even care about understanding the rates of photosynthesis and cellular respiration in some sort of algal cell. So if you're in the Pacific Northwest, of course, like the Hood River or Hood Canal is a fantastic one to look at. If you're in Southern California, there is the Salton Sea. I am less well-versed with my East Coast ones. But if you are really curious about finding one, send me an email. I'm sure I can dig one out for you in the area that you're in. So another context that you could also do if you're like, ecology, not my thing, you could talk about actually just doing like food production at home. So I used to work in a biohacker space and we had somebody that was trying to make these aquaponic systems. And you've got fish on the bottom in a big kind of tank, and then you've got plants on top, and you're basically circulating water to kind of bring those nutrients up to the top for the plants. And after a couple of months of this guy's system running, kept coming in the morning and there'd be dead fish at the top. And we're like, what's going on? It wasn't really a well-sealed building, so we thought maybe something was getting in there like a raccoon and trying to fish around and grab them, and was just, hurting a couple of fish. But then he set up a camera to actually see if there was an organism coming in trying to get his fish. And what he saw was at like 3 in the morning, all the fish would come from the bottom of the tank and they'd run to the top of the tank. And so what we think was going on was there was a hypoxic event taking place because he was using basically a giant clear aquarium tank to do this. And it definitely had, some like algae you could see just encrusting on the walls. And so the idea we think what was going on was that you were having an O2 drop during the night because all those algal cells now are doing that, and the pump wasn't running at night as strongly as it was during the day. And so those fish are coming to the surface to try and get oxygen, and then some of them just based upon size, body mass, were not making it. So there's a couple different ways that you can use to kind of draw students in from a context point of view to then get them into the heart of this lab. Once you get into this lab, there's a whole series of different lab activities you can do with the students. The first one Bio-Rad has you do is just show them that the pH indicator works by having them kind of blow into a cup of it. And basically what they'll see is this thing that kind of goes from being, this orange color at atmospheric CO2, as they're blowing CO2 into it, it becomes more yellow. If I'm trying to run this with multiple classes, so if I'm trying to stretch this one kit over like say 5 sections, this is the one or actually there's two things I skip. I would skip this because this just blows through a bunch of pH indicator, and I'd rather save that for kids to do rate studies with. And I also would skip the microscopy side just from a timing point of view. So we do have what we call a debeading solution that comes in the kit. So you can actually dissolve out the beads and get the algal cells out and then put them underneath a microscope for students to look at. And unless you have students that are already like really into microscopy, I don't feel like this is a big plus for them, just because they're just seeing small green cells. There's nothing super, super entertaining about them. So a lot of times I'll kind of put those two activities on the back burner and then focus in on just doing a light dark reaction with students, and then scaffolding that information that they learn to have them look at different variables. So we already talked about the pH indicator and how the system works. So let me walk you through how kind of the core lab of this looks like. And this is the light dark reaction. So what students will do is they will basically be given 2 cuvettes. They'll be given some of this pH indicator solution, and then about 20 to 25 beads. Students will basically move those beads into the cuvette so that there's 10 per cuvette. They'll wash the beads with water just to remove any leftover kind of indicator or storage solution that's on the beads. And then what they'll do is they will add 1 milliliter of indicator to these cuvettes. And so the big thing to think about here is this is a ratio that if you are modifying this protocol at all, you want to maintain. So you have 10 beads per 1 milliliter of indicator, which means you have basically 0.1 mil per each bead. And so that's kind of what you want to carry around if you decide to play around in, say, different size containers, things like that. If you maintain that ratio, you'll see about the same, getting to the end of that spectrum in about 15 minutes for both of those. So once the indicator's in there, I have the students kind of mix it up just to make sure that pH indicator is well saturated into the beads before they start. There's little tops that come with these cuvettes, so you can clip those tops on so that way they don't leak. And then what's going to happen is one of those cuvettes is going to go in the light, and then one of those is going to go into the dark. So the easiest way to make a dark cuvette is just to give students a little aluminum foil square, and what they can do is they can just wrap up that cuvette in that. And the reason I like to use the aluminum foil square instead of like having to put it in a desk drawer or something like that is if I really want to just monitor the difference between the light reaction and the dark reaction, I want to make sure these cuvettes are getting the, as much as possible, the exact same experience except for that one variable. And so I'll have them foil up that one cuvette and usually lay it side by side with the one that's going to be exposed to light, and then slide underneath my light source. So that way, if there's any heat or anything like that emanating from the light, I will also be picking that up in the dark sample. And so that way we don't have an uncontrolled variable there. For lights, there's a lot of options now out there on the market. The one thing I would suggest is going LED. And that's mainly because most of you folks, if you've been doing photosynthesis labs for a while, you're using those like bright halogen lamps that are really hot, and you usually are having to use a heat sink in between them, stuff like that. You really want to use something that's LED that you can get a lot lower basically heat transfer off of. And a lot of these LEDs now will produce just in the wavelengths that you want. And so you can actually buy LEDs, right, that are just for growing plants. You buy those, and those are like the best thing you can get for this. And the beauty is a lot of those, they pull a lot less energy. And so you could even just buy these ones that like clip onto battery packs, those like little USB battery packs, and just have those at each of the desks. Or if you don't have any lights at all and your students come in with cell phones, you can actually use the LED on their cell phone. It's a little more blue skewed. And so it doesn't go quite as fast as one of these ones that's been engineered, obviously, for plants, but you can still get the beads to kind of, do change in photosynthesis. It'll take maybe about 20 minutes though, instead of 15. And all you do is basically have them turn on their light, and they will place the bead right, or the cuvette right on top of the light. And that'll be enough for it to kind of change in that period of time. So students would basically record what the pH looks like either qualitatively or quantitatively. So since it's in that cuvette and those beads are basically keeping it so those algal cells aren't floating all around, you can drop this in a spectrophotometer, shoot a beam path through here at 550 nanometers, and pick up basically an objective value that will allow you to really closely monitor this rate over these 15 minutes. So traditionally we have students measure them at 0 minutes, 5 minutes, 10 minutes, 15 minutes, and that's enough for them to be able to kind of graph it out and get an idea of what the rate of photosynthesis is and cellular respiration for this kind of first condition. That's really important because I feel like this is the scaffolding portion of the lab. This is where I'm at every desk, making sure everybody is running smoothly with it, because after this experiment, I'm going to cut them loose to kind of do a little bit of their own experimental design and allow them to kind of maybe go after variables that maybe I didn't even think about. I usually will hand out a couple of variables to them just to get them thinking, which is the fact that they could change the light intensity, right? You could just move that light higher away from your source or closer to your cuvettes. You could screen the light, so you could use filters, right, to get to specific wavelengths. Or, I mean, Amazon has super cheap LEDs in very specific wavelengths now. You could just buy a set of different wavelength lights and be able to kind of clip those in, have them use them. You could also have them modulate the temperature. And this is actually one that I love to kind of like start off with, especially if I'm doing that kind of ecology context with them, because once they've seen that rate and they go, okay, cellular respiration is moving this fast, photosynthesis is moving this fast, I say, well, what happens if now surface water temperatures change? And does photosynthesis and cellular respiration, does their rates, do they change in lockstep? What happens if they don't? What happens if, as sea surface temperature raises, cellular respiration moves much faster than photosynthesis? What's that mean for overall carbon cycling, things like that? And so there can be some big kind of like, ooh, yeah, things could go really bad. Why don't you guys test that? Make sure that's not going to happen. And they can do that by doing this kind of temperature variable with the system. You can also set up little mini ecosystems. So you could put like those snails you can find at aquarium shops in there. And what you'll see then is like, you'll have a really hard time ever getting that system to go purple again because you've got a heavy breather in there. A lot of places too, if you don't have like an aquarium shop, if you go and you tumble a lot of logs in a forest, or even just like large rocks in urban areas, you can find these little tiny, what they call glass snails, and those guys will survive in these systems perfectly fine too. So you can make these little ecosystems with them. One thing to know is that this lab we originally designed for AP biology, and that's why it's in the cuvettes, and we planned on having you use potentially a spectrophotometer for it. But we also made another version of this lab, realizing that a lot of people were using this qualitatively. So the kit comes with a little color scale, and so students can just qualitatively look at that color change against that scale and then make estimates about what the rate change is that way. So there is a second basically version of this kit where instead of doing it in cuvettes, you do it in PCR tubes, which are just these really tiny little microcentrifuge tubes, which then allows you to take the number of beads that were in the kit and spread it out over more stations. And so if you're looking to get basically more stations and not do it quantitatively, Bio-Rad's photosynthesis general biology kit is the way to go. But if you're trying to do that qualitative stuff, definitely do the one with the cuvettes. So we worked with Vernier with this lab a bit to make sure that this system works on their spectrophotometers. And so they've got an entire kind of write-up on using this in their spectrophotometer. One thing I would stress though, with the spectrophotometer, you always want to know where your beam path is, because I've definitely had a couple people will write us back and be like, hey, I tried to use this and I'm not seeing any change. What's going on? And what happens is their cuvette was just such that when you fill up 1 milliliter of indicator, the beam path is just going over the top of it. So they're just measuring the air every time. So what you want to do is figure out where your beam path is in your spectrophotometer before getting started with this. And you can do that just by continually adding more of the pH indicator until you see basically a change at 550 nanometers and you go, okay, that's the level I need to be at. And so then you can just scale the number of beads in order to hit that volume, and you're kind of ready to roll with it. Since I've used this lab for quite a few years, what I'm going to do is I'm going to show you also some of my tips and tricks for when I use this with multiple lab groups. And that's mainly because when we first wrote the protocol for this, right, it got beta tested a handful of times, and then over a number of years, you start to find other kind of tricks that make it a little bit easier to pull off this for like, say, if you're doing like the whole multiple classroom thing I was talking about. So let me pull up my tips and tricks slide deck. I'm going to just show you guys a handful of those, and then we can get into doing some questions. So hopefully you guys can see that. All right. So for the prep of this lab, it's actually, from a Bio-Rad point of view, this is the easiest lab Bio-Rad has to prep. And it's mainly because all you're doing is washing these beads off after they've come out of their kind of storage solution. And you're just doing that with distilled water. And then you're going to make the 1x indicator from a 10x concentrate. So you're basically adding distilled water in there in order to bring that up to a 1x solution. Bio-Rad's protocol, though, has you take that 1x solution and set it on your desk for 2 to 3 days for it to equilibrate to atmospheric CO2. And the way that you can tell is it will stop changing colors. I'm a very impatient person. And so I was like, I can't 2 or 3 days. That's, oh, no. It's a surface area problem. And so what you can do is you can make up your 1x indicator, get yourself 2 cups, and just start pouring it back and forth. And if you do this, usually it takes about 2 to 3 minutes. What you'll see is the color will slowly start to change, and then all of a sudden it will stop changing. You're now equilibrated to atmospheric pH or atmospheric CO2, and you can kind of move on to the next step. The beads need to be activated the night before using them. And what this means is you've had these organisms that have basically been sleeping in your fridge for months and potentially ours for months. So they haven't had any food in a really, really long time. So they don't have a lot of their kind of storage materials up and ready to run. And so what you want to do is take those beads, wash them in distilled water, and then I like to put mine in the pH indicator just so I can see how well they're functioning. So what I'll do is I'll add them basically to just a Petri dish. I'll pour on some of my 1x indicator, and then I'll put them underneath a light source overnight. And what'll happen is I'll see that pH indicator in there slowly shift purple. And that tells me, okay, you know what? They're all up and functioning now. They're working well. And so that way when I hand them off to the students, they will have like the highest rate possible. If you only have those really hot lamps, what you really want to do here is make sure you give yourself a nice kind of heat sink source for this. So the picture on the bottom right there is just one of those. I've got a giant liter of water that's sitting on top of my Petri dish with my hot lamp source up above. But really, like I said, buy yourself some LEDs and you'll never go back. They make all the difference in this one. For the transfer of the beads, there's 2 ways to do it. And there's a picture of one of them at the bottom left here. So there's still great debate internally at Bio-Rad on how best to transfer beads. Some people like to take these transfer pipettes, and you get a ton of them in the kit. And they like to cut them kind of at a side angle so that you have a scoop, so you can scoop up beads and move them. I personally am a sucker. So what I'll like to do is take that transfer pipette, cut it perpendicular to the barrel. And now that barrel is just barely bigger than the beads, and I can just kind of vacuum them up, which makes it really easy to count them. Bio-Rad's protocol has you delivering, 20 to 25 beads in just a microcentrifuge tube to the students to have them then take out 10 and put it into each cuvette. I find that to be kind of a waste of time because I'm already putting my time in moving beads. And so if I just move them directly into the cuvettes for the students, it saves me an extra 5 to 10 minutes, depending upon the students, once we get into the classroom. So I'll just immediately move mine right to the cuvettes and not follow the Bio-Rad protocol on that. And the reason I also do that is it makes it so that I can set up a self-service station. So for Bio-Rad's AP Biology kit, it comes with all these cuvettes and you get this giant rack, and you're going to use about half of those cuvettes for how the protocol is written. So what I'll do is I'll put my 2 cuvettes that I made for each student group in a row for light and dark. And then I'll actually just put all the reagents they need also in cuvettes that I'm not going to use otherwise. So I'll make a cuvette of pH, I'll make a cuvette of water, and then I'll give them a cuvette for waste. And so students basically come up, they just grab each of those 5 cuvettes, they grab a transfer pipette from the Bio-Rad box, they go back to their desk, and they've got pretty much everything they need to run the lab. I'll also have the color cards up there. So these are the color cards. First time you buy the kit, cut them out, put them in Ziploc bags, never buy the full kit again. Just buy the refill kit every year, because at that point you'll have the color cards, you'll have enough transfer pipettes, to do this over and over. And all you really need every year to run this will be the beads and the indicator solution. Yeah.
So we're about 10 minutes until the end of the session. I do want to open it up for questions and see if there's more we want to talk about just from the people here. We got one question from the chat specifically to talk about extensions. So you went over that really nicely. And I just want to commend you for everybody. So Damon was not planning on talking here. The audio kind of crapped out and he just stepped up in this. So it was a really nice talk. Well done.
Let me nail out these two things, George, and then let's switch to the questions. I think
sounds great.
These two trips are really helpful. So if you're doing a qualitative or quantitative version of this, I really encourage you to make some pH buffer visuals for the students. And that's because the color card's great, sticking something in a spectrophotometer is great, and getting that quantitative number. But for students, it really helps them, I feel like, to see something that looks exactly like the thing they're going to be working with and be like, okay, that's what it looks like. And these are also going to be great if you use a spectrophotometer. So what I'll do is I'll add pH indicator to 3 cuvettes, a drop or two of vinegar into the far left one there, they get it bright yellow, and then a little spritz of Windex in that far one, and that'll take you all the way out the other direction, going more basic. So that way you can drop these in the spectrophotometer and also show them the full end of the spectrum and the center of the spectrum. You could use this basically as a standard curve. The other thing I wanted to show you that I think is really important, because I'm sure if I were to ask you guys how many of you guys have a spectrophotometer, it would be 5–10% of you. You can kind of hack a spectrophotometer by using a color dropper tool like what used to be in Photoshop. Now there's a ton of free ones online. My favorite one is Color Cop, which just sits on top of Chrome. And so what you can do is you can use this kind of like a spec. If you hover your color dropper over basically pixels, it will give you a quantitative value for the red channel, the green channel, the blue channel. And using this lab, like I did that with this, and I can get a standard curve that students can work with. I can then have them look at their unknowns and then plug those values into that standard curve. You're of course never going to publish with this, but it gets them those mathematical skills, and you didn't have to pay for a spectrophotometer, and every student has one. There's no giant backlog of people waiting to get on the spec. So those are like just two tips I think are well worth pointing out. Let's go ahead and open it for questions. Since there's so few of you, feel free to unmute yourselves. Actually, I'm just gonna go ahead and unmute you guys if you don't mind. And that way, if you've got questions, we can just kind of handle them out in the open. And then we've got, I think, about, five or so minutes to kind of do that.
So you did a nice job talking about extensions. What's really wonderful about this kit is how robust it is. And CTSs like Damon, they take a lot of ways to go and basically break our kits. And you can have your students do the exact same thing. And you can do really nice experimental design. So what Damon was talking about, just to give you a behind the scenes thing, we were testing cuvettes and I had the exact same problem he was talking about. How do you actually do the measurements using a spectrophotometer? Because I had to be very precise and I had to come up with this experimental design of how to, use these cuvettes, get the right wavelength measurement working. And that's something that you have to do in real proper experimental design, and you teach your students doing that with these kits. So it's really nice stuff like that. So I don't know what else you have, wavelength, color of light, distance, the activation of these beads. You have the students do the activation because they have to revive these beads.
And another one that you can do that is not recommended unless you're at the end of using it with all of your groups of kids is finding, I come from industry originally, so I love to see failure. And I know students hate to see failure. I want to know, when do those cells die, right? At what temperature do they stop being functional? Because that's a really important thing to know about that system. And so that's something I would do with my last group of students, or you could do it with just 1 or 2 beads in a really small container and figure out where is it that those organisms stop being able to tolerate heat. And for alpha cells, it will be surprising at how low that is.
Do you have different types of algae available?
We don't. We only are working with Synodesmus obliquus, and that's mainly because we went through man, I want to say almost like 10 different species, like commonly cultivated ones. And this is the one that basically put up with abuse best. And so that's why we kind of settled on using this for this lab.
All right.
Yeah.
Do you have any plans to do zooplankton next?
I have not heard that in the works, but if you get a good suggestion for us on that one, send me a couple of species. I can always pitch that to the marketing folks. But we'll see how receptive they are.
The relationship is so it's sort of elusive or something, you know, but it sometimes I think it looks like there's a relationship between harmful algal blooms and zooplankton migrations or zooplankton populations.
Yep, and
migration and population.
I see that too. Where I live by like a brackish water space, and then we definitely see that we'll have a big algal bloom this heart of the summer, and then we get all of our zooplankton bloom usually like now, like October through about now.
Yeah. Yeah, they're definitely together, stuck together somehow.
Yeah.
So it has to be a big, that's a big deal, I think, especially for the future with like ocean acidification and a lot of the things that you were talking about. I'm also kind of interested in a little bit about like how not just the increased temperature, but sometimes it seems like there's an increased exposure to UV light, and then the UV light maximizes the photosynthesis of the algae, and then, you know, what's going on with that with the adjusted pH too. So I think kids could really figure that out though, because they don't have the, you know, like I remember a bunch of stuff, man, they don't remember anything. So
well, the cool thing about this is that you do have enough of that pH indicator that if you have access to a handful of those organisms, or even if you've got like that wetland around, you could try just squirting some of that into the pH indicator and then exposing it to different light to see if you can see effects from that. Like I've definitely tried putting other plants in there before, and I can get some of those to do rate changes that are a lot slower than the algal beads, but you can use this pH indicator system really well with other live organisms, which is kind of fun.
Oh yeah, that'd be great to do with kelp and pseudonychia together, not separate, but together.
But together, yeah.
To see those changes in pH, and you know, I mean, the temperature naturally changes with the pH, but really to look at that, because there's some symbiosis is some of the factors that we might be missing.
Seaweed that doesn't have pseudonychia all over.
Yeah, yeah.
Just having to strip those off first somehow, maybe with the salt or the soap or something.
Yeah, yeah. But thanks for your talk. I got a lot out of it. Especially, I really love your spectro hack there. That's really cool.
Yeah.
I like
it's
that
helpful.
kind of stuff.
Yeah. Well, I'm glad you enjoyed it. Thanks. Sorry about the audio at the beginning. I'm not sure what was going on when that video wasn't playing the audio right.
Oh, I was late.
Oh, okay. You missed that part. All right.
Yeah.
Called the cyber black hole.
Yeah, right.
Dave, do you have the survey link to add? Do you have the survey link to add?
I do not. In the video, but then it wasn't in the end of the video, so I was gonna be digging that out instead of giving the talk.
So
I think it is here. You know what? I'm just gonna do this. I think it's just NSTA. Is it 2020? And this one is probably workshop like 8. Let's see if that works. Let's see if that link works. Nope. Ignore that.
I'll see if I can get that. Damon, can you go ahead and answer a question that we have, which is about how do you prepare and store the beads for reuse?
Yep. So for storage and reuse, so I'm using that kind of hacked version of the kit that you showed, where I organize everything in the styrofoam box the cuvettes come in. So I literally had students come back through and re-rack all of those cuvettes right where they took them and leave their beads in the pH indicator. And so I'll just take that box now and then put it someplace in my classroom that there's ambient light, but not direct light, because I don't want them to get hot. And those beads will just continue to do a mild level of photosynthesis in the background, cellular respiration. And so after activating, they easily stay alive for a month in your classroom. And then next time the kids go to work with them, next student group will just go through, they'll pull out those 5 cuvettes again. Their beads this time will have the pH indicator in there. So what they'll need to do is they'll basically remove that, wash it with water to get it back down to kind of, atmospheric CO2 again. And then the whole process just works. And so that's how I store them. I'm trying a long-term storage project right now at home where I'm taking that cuvette box and putting it in my fridge and then only taking it out once every 3 months for a couple of days to give it light to see how long I can get them to go. Because I had somebody report that they had their beads that they used for 2 years in a row. And I was like, I don't think that's possible, but I'm going to try. So I'm at month 8 right now on my set. And they're doing okay, but I think I'm starting to see a little bit of, fade. So I wouldn't recommend that. But if you're just doing it within a month period, just put them on the desk in place where they're getting ambient light in that cuvette box, and you'll be good to go.
Yeah, I've added the link there. This is the NSTA 3 link.
Okay, thanks, George. So with that link, it gives us a little bit of information about kind of what you guys need. It helps us kind of design new things we're doing. And then the sweetener for you guys is there's the chance to win a couple prizes with it, including one of our biotechnology textbooks, and then also a potential kit.
Oh, cool. I know researchers who have needs. All right, thank you for this presentation. It was really good.
Yep, you got it.